आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. In this podcast, we welcome Dr. Anupama Ghosh, who have associated with Bose Institute for almost 10 years. We will listen to her wide work experience in her own words.
I am Anupama Ghosh. I am associated with Bose Institute for about 10 years now. And I started my scientific career as a microbiologist. I did my PhD in the field of molecular parasitology, and then I moved on to work with plant-microbe interaction and in my postdoctoral studies. So in my postdoctoral studies, I was introduced to plant-microbe interaction, specifically to the pathogen effector proteins or pathogen effector biology. Now, let me tell you what are pathogen effector proteins and why they are important in plant microbe interaction. So, when a plant is infected by a microbe, it may be a bacteria or a fungal cell, there is a communication between the plant and the microbe. And pathogen effector proteins are they are not always proteins, pathogen effectors. They are the molecules that the pathogen release into the interface between the pathogen and the plant. And those molecules actually control the host, or in this case, the plant defense responses. So we tried to understand how these proteins or these effectors actually take control over the host plant. So once I returned here and I did my postdoctoral research from Max Planck Institute under the supervision of someone who is a stalwart in this area. And so I learned Eustallago maydis effector biology from there. And Eustallago maydis is a basidiomycete plant pathogen. So that meaning it is a biotrophic pathogen. So when it infects the plant, it keeps the plant alive and it does not kill it. And so it has this need to switch off the plant's defense response up to a certain point. So it gives us an ample opportunity to study how the plant actually defends itself. And so when we study how the fungus uses its proteins to get control over the plant, it gives us an opportunity to also know how the plant defends itself. So we have studied many different proteins that are involved in establishing the infection and how they are contributing towards the disease progression. So the idea is we want to know how these effector proteins are, or what are the targets of these effector proteins. And once we know that, we can design ways to modify plants so as to make it resistant against pathogen invasion.
Dr. Anupama Ghosh speaks about the range of vegetables and crops on which the experiment is done, and also about the results.
More recently, we are the first ones to show that to profile the RNA or the nucleic acid also present in the plant apoplast, and they have a different profile while infected by, maize when it is infected by Eustallago maydis. And we have profiled that in collaboration with, we have some national as well as international collaborations on them.
Dr. Ghosh explains in details about the research collaborations.
So we have collaborations in NIPGR in India and also from United States. So recently I have been awarded with a Fulbright Fellowship to conduct a research in collaboration with Indiana University. And so we have very exciting results which are yet to come out. So we are working on that. And I'm hopeful that we'll be getting interesting information that we can translate into crop improvement.
Her work on plant microbe interactions to pathogens has an effective application for the farmers as well as for the industry. Her valuable words will help us understand that how the industry will get the benefits from it.
So till now we have been, as I already said, I was working, our lab works on Eustallago maydis. So this is a very specific pathogen and it infects maize. So we are currently working on the defense response mechanisms of maize and the virulence mechanisms of Eustallago maydis. So, but Eustallago maydis, definitely it has some specific mechanisms, but there are some which are common to other pathogens which are not that, Eustallago maydis infection is not that harmful, but there are pathogens which are more harmful than Eustallago maydis. So once we know the mechanism, how it is, because it is easy to study Eustallago maydis maize because you can keep the plant alive for quite some time. So you have enough time to study those defense responses. And so once we know that, we can actually utilize those information for other pathogens which are more harmful than Eustallago maydis. And that will give us an idea about the plant defense responses. And more recently, we also started working on some extracellular vesicles. So these are small packets, you can say, which are secreted by the fungus as well as the plant. And these packets are filled with biological macromolecules, including proteins and nucleic acids. And now working on the hypothesis, and also there are many other laboratories worldwide who are working on the hypothesis that these packets are actually the carrier of information from one cell to another. So if these packets can be targeted, these extracellular vesicles can be targeted for improving the defense response of the host plants. So we can design strategies, but these are all basic research till now, and that needs to be, we need to work further to know more on that and to apply those in the translational research. So we need to know what are the mechanisms and all. And if we know that, then we can definitely we can design crop plants which can utilize these strategies to defend itself against diseases, plant diseases, pathogen infection, microbial infection.
We are thankful to Dr. Anupama Ghosh for providing us with her valuable time.